Hello and welcome to the Washington University in St. Louis Evolution, Ecology, and Population Biology and Living Earth Collaborative Seminar. It's my pleasure to introduce today's seminar speaker, Rachel Becknell. Rachel is a sixth-year PhD student in the Evolution, Ecology, and Population Biology program here at Washington University in St. Louis. Rachel received a bachelor's degree in Spanish and International Studies from Westminster College in 2008. And she then went on to obtain a second bachelor's degree, this time in biology, from the University of Missouri in St. Louis in the year 2015. As an undergraduate, she was already highly involved in ecological restoration research, including through REU programs, research experience for undergrads programs at the University of Montana and at the Missouri Botanical Garden, and through a senior thesis research project with Matthew Albrecht at the Missouri Botanical Garden. The results of her senior thesis research on how shade affects growth of the federally endangered legume Astragalus bibullatus have already been used by the Tennessee Department of Conservation and Missouri Botanical Garden for the selection and preparation of sites prior to reintroduction of this endangered plant. So her research has already benefited ecological restoration practices. And one of her dissertation chapters that she's going to tell us about today investigates the effects of soil microbial communities on that same legume species. The core of Rachel's dissertation research was developed in the lab of Scott Mangan, who is now an associate professor at Arkansas State University. In her research, Rachel asks questions that are at the frontiers of our understanding of how soil microbial communities interact with plant communities and what the outcomes are for plant community diversity. And something that really sets her apart in her research is how she always has this keen emphasis on how her results will apply to ecological restoration practices. So really translating from basic to applied sciences. Her projects are always impressively large in scale, whether in the greenhouse or in the field, and she has extensive experience mentoring undergraduate and high school students who have been involved in these projects, including through her leadership of student research teams at Tyson Research Center. Rachel has been very successful in securing funding for her dissertation projects from several different entities, including from the Garden Club of America, the Missouri Native Plant Society, and the Webster Groves Nature Study Society. And in addition to all of these research and mentoring accomplishments, Rachel has been highly involved in community service from the time she was an undergraduate to today. She was the president of the St. Louis chapter of the Missouri Master Naturalist for 2 years as a graduate student. And when she's not out performing her own experiments to study specific hypotheses about best practices in ecological restoration, she spends her free time helping clear out invasive species at Shaw Nature Reserve and other local natural areas. Rachel is also passionate about effective science communication, and she has presented her research at several local venues, including at the Missouri Native Plant Society and the Webster Groves Nature Study Society, in addition to presenting her research to scientific audiences, including at the 9th Annual St. Louis Ecology, Evolution, and Conservation Retreat in 2019, and speaking with us here today at the LEC EEPB seminar. So just before Rachel begins her talk today, I want to remind the LEC Biodiversity Fellows to please join us for a reception over Zoom at the end of the questions following her talk, and you should have received a separate Zoom link via email. So now it's my pleasure to hand over the Zoom microphone to Rachel Becknell, and we're very excited to hear about your really exciting research.
Thanks for that awesome introduction, Rachel. I appreciate it. I'm excited to be here today. So starting off, I wanted to introduce the motivation behind all of my graduate research. I care a lot about using ecological theory to better inform the practice of ecological restoration. But in turn, those who study ecological theory can learn a lot from the practice of ecological restoration. And this isn't a new concept. Tony Bradshaw, back in 1983, he wrote that in order for us to be able to show that we truly understand how ecosystems come together, we need to be able to put them together in practice and show how they work. So for my talk today, I'm going to talk about 3 of the major projects in my dissertation. I'm going to start out a little bit more broad and talk about the effects of soil microbes on tallgrass prairie species. And this is a photo of that first experiment that I did back in 2015. And then I will get a little bit more specific and talk about the effects of soil microbes and plant community relatedness on the growth and establishment of hard to establish prairie plant species. And finally, I will talk about the effects of soil microbes on the growth of the federally endangered Astragalus bibullatus, otherwise known as Pines ground plum. Okay, so starting out, I need to talk a little bit about some theory and niche partitioning in particular, leading to coexistence. And it's really important for us to understand the factors and the mechanisms behind coexistence, especially in ecosystems such as the tallgrass prairie that can have 200 plus species in one prairie. So it's very important to understand how so many species can coexist in one space if we want to be able to put these ecosystems together. So in this example, the classic explanation for how many different plant species can coexist in one space is that of niche partitioning. In this example, you see species 1, 2, and 3. And species 1 and 2, they're both in the aster family. So they share a similar niche. You see that they have a similar rooting structure, whereas species 3, it's in the mint family. Its roots are much longer. So in this situation, species 1 and 2 are probably better at taking up soil nitrogen, whereas species 3 is probably better at taking up soil moisture than the other 2 species. So according to niche partitioning, in a circumstance where species 1 and 3 or species 2 and 3 are competing with each other, they are likely to be able to coexist because they fill different niches. Whereas if you have species 1 and 2, because they're more closely related, they share more similar niches, the outcome is a little bit more uncertain, and it's likely that one of those species would outcompete the other. But another less studied reason for many different plant species being able to coexist in one space is that of soil microbes, and in particular, the impact of soil pathogens. In this example, we have species 1 and 2. 1 is an aster and 2 is a mint species. And both species have soil pathogens that specifically like to attack the roots of that species. So you see species 1 has these red pathogens that are accumulating underneath the roots of that species. And the further away you get from it, there are these pathogens are less abundant. And the same is true for species 2. It has these green pathogens and they become less abundant the further away you get. And this has an impact on each species' offspring. So species 1, you can see that close to the parent plant, there aren't many seedlings that are able to grow. And the further away you get from that parent plant, the more seedlings you have. And seedlings in particular are very affected by soil pathogens. And the same is true for species 2. The further away you get from the adult plant, the more of its offspring are able to survive. And in this circumstance, we would call this negative plant soil feedbacks. And this should be able to lead to coexistence of each species, because each individual species suppresses the growth of their own offspring. So over time, you would expect for that to lead to coexistence. So you see on the x-axis, I have time, and on the y-axis, I have population density. The blue line is the growth, the population density of species 1 over time, and the red line is the population density of species 2. At time 1, you see that species 1 has a much more dense population than species 2. So what you would expect is that the pathogens in the soil underneath species 1 would also be very dense in attacking the species offspring. Whereas for species 2, because it's not as dense in that community, you expect for it to not have very many pathogens. So its population numbers are able to increase, but species one's population density would be decreasing over time, so that at time 2, it has swapped so that now species 2 has a much more dense population. And it's this fluctuation in the population densities of these 2 plant species over time that should allow for them to be able to coexist. In my first experiment, I studied the effects of soil microbes on 18 different tallgrass prairie species. These are 6 of them that I worked with. And the first objective for this study was to determine the potential for negative plant soil feedbacks to structure these tallgrass prairie communities. The second objective was to determine whether plant evolutionary history or plant relatedness affects the strength of these feedbacks. And finally, I wanted to use DNA metabarcoding to examine whether there is evidence for specificity of the soil microbial community. So my experiment was in 2 phases. And in the first phase, I collected soil from a reconstructed prairie at Shaw Nature Reserve. And then I planted seedlings of each of the 18 species in their own pots and added some of this soil to the pots. Allowed them to grow for 9 months. After those 9 months, the soil underneath these species should contain more of those species-specific soil microbes. So in the case of soil pathogens impacting the species a great deal, these pots should contain a lot more of those species-specific soil pathogens. In the second phase, I took the soil from those pots along with the fine roots, and I added it to new pots and planted new seedlings of each species. So each species got planted in its own soil with 8 replicates, but it also got planted in the soil of the 17 other species. So this was a fully reciprocal design. So each species was planted in all of the other species' soil. And it was also done in live and sterile soil so that we would be able to disentangle the impacts of microbes from abiotic effects. All right, so in order to determine the strength and the direction of these plant soil feedbacks, we need to be able to utilize the growth of our plant species from our experiment. So in this example, you see a yellow species and the green species, keep it simple. The x-axis is their soil biota that they grow in, and then the y-axis is the seedling growth. In this example, the yellow species grows much smaller in its own soil than it does in the soil from the green species. And the same is true for the green species. It grows much smaller in its own soil than it does in the soil of the yellow species. So each species is essentially escaping its own species-specific soil pathogens when it's growing in the soil of the other species. So to calculate these feedbacks, we need to add together the growth of each species in their own soil or their home soil, and then also add together the growth of each species in the away soil and subtract that from their growth in the home soil. In this circumstance, because each species grew much larger in the away soil, you end up with a negative interaction. So I did end up finding that this was an incredibly prevalent interaction in my system. In these results here, we're looking at Monarda fistulosa and Rudbeckia hirta, and they displayed negative pairwise feedbacks when grown in their own and each other's soil. And this is a circumstance that should lead to coexistence between these 2 species whenever they're growing together. But what's really important is that whenever they were grown in sterile soil without microbes, this relationship completely disappeared. So this shows that these negative feedbacks was driven by the soil microbes. But overall, for all 18 species, I found that negative feedbacks were incredibly prevalent. This is a figure showing the average feedbacks. So if you look at the first species shown there, AG, that's Andropogon girardii, a common grass in prairies. And the average feedback is calculated by just taking an average of all of the pairwise interactions it has with the 17 other species in the experiment. So you see that when microbes are present, negative feedbacks are incredibly prevalent, but that completely breaks down whenever they're grown without microbes in the system. So my second objective was to determine whether plant evolutionary history affects the strength of plant soil feedbacks. So you recall from this example that negative feedback occurs whenever a plant is more affected by its own soil microbes than it is by the other species. And we expect for this to occur whenever two plant species are more distantly related from each other, because we would expect for them to not share the same soil pathogens and be able to escape from them when they're growing in the other species' soil. So whenever species are closely related, expect for them to share a more similar pathogen community. So when they're growing in the soil of the other species, we shouldn't see any difference in their growth, and this would be a neutral interaction. So I expect to see the more distantly related species to have a much stronger negative feedback. So only in the live soil, but then in the sterile soil, I expect to not see any phylogenetic signal at all. And that is exactly what I saw. I found that the more distantly related plant species had much stronger negative pairwise feedbacks than those species that were more closely related to each other. But this was only true when they were growing in the live soil. So finally, for this project, I utilized DNA metabarcoding to examine whether there was evidence for specificity of the soil fungal community. And this work was done in Dr. Christy Edwards' lab at the Missouri Botanical Garden. So for feedbacks to be able to occur, these soil microbial communities, they must differ across the plant species. The first steps were to collect the soil from the roots, around the roots of each of the species. We extracted the DNA from the soil, and then we isolated the fungal specific primers. And so we focused on fungi in particular because there's a lot of evidence out there showing that fungi are a very prevalent pathogen in this system. And then I performed bioinformatics to determine whether, the microbial community similarity. So I found that fungal communities do differ significantly among the plant species. So each of these dots on this NMDS represent a sample. So you'll see that agar, Andropogon girardii, has 3 samples. And you notice that for most of these species, they're clustering together, showing that their soil microbial communities in each of the samples are very similar and distinct. I got a little carried away with my ovals. Okay, so we found strong evidence that negative feedbacks are prevalent among tallgrass prairie plant species. And because we had 18 species in this experiment, we're better able to generalize about what is going on in the system. Many of these plant soil feedback experiments have 5 or less species in them because it does take a lot of work and space to be able to do them. We also found a strong phylogenetic signature with the more distantly related plant species having much stronger negative feedbacks. And finally, I found that plant species do possess a distinct soil fungal community. Okay, I'm just gonna. Okay, so my next project is getting a little bit more specific, and I'm looking at the effects of soil microbes on hard to establish prairie plant species. And I performed this in 2 research gardens at Washington University and St. Louis's Tyson Research Center. And my first objective was to determine whether soil microbial origin is important for the growth of hard to establish tallgrass prairie plant species. And by hard to establish, I mean tallgrass prairie plant species that restoration practitioners have a difficult time getting to grow in restorations. I also wanted to determine the role of evolutionary history or plant relatedness on the growth of these hard to establish plant species. And I wanted to tease apart the contributions of soil microbes from that of competition. And this experiment was performed in communities. I wanted to point out that my previous experiment was performed only with individual plants. And it is very important to scale that up to look at communities. So I looked at 4 different hard to establish prairie plant species, and I selected these plant species after consulting with various ecological restoration practitioners, the members of the Missouri Native Plant Society, and asking them which species they knew people had a hard time getting to grow in ecological restorations. And I wanted to know how these hard to establish species grow whenever they're surrounded by a background community of species that are much more common in tallgrass prairies. So I had 16 different background species, and these were from the original greenhouse experiment that I performed. But I do want to point out that 2 of the species that were in that original experiment, Allium stellatum and Oenothera macrocarpa, I removed those from this experiment because they are more of a glade species, and I wanted to focus specifically on tallgrass prairie species. So this is an aerial view of one of the research gardens at Tyson. The green squares are 1 meter by 1 meter plots. So I organized them in groups of 3, and one plot within the group has seedlings that have been inoculated with soil from remnant prairie. The remnant prairie is Tucker Prairie outside of Columbia, Missouri. And the old field was an old field from Shaw Nature Reserve. And then the sterile treatment was a mixture of the remnant and the old field soils that I autoclaved twice. Within each of these plots, all 4 of these hard to establish species have been planted. But I also planted 6 of the background species, and these were selected based on their community relatedness. So some of these groups have plants that are more distantly related to these hard to establish species. Some are more closely related, and then there's a spectrum of plots that are in between as far as how related they are to the hard to establish species. So first, I rented a mini excavator. It was very fun to learn how to drive that. Dug the holes, filled them with these plastic liners to prevent the roots from escaping these plots. And then I had to sterilize all of the soil that I pulled out of these plots. This is a soil pasteurization system. The green wagon in the back has this grate with holes in it in the bottom, and then we fill it with soil. This is a steam generator in the front here, and it pumps steam into the wagon, and the steam flows up through the soil and sterilizes the soil for me. So we had to run that through twice. This is a fun video with the process of filling them. I think everyone knows whenever you start an experiment like this, you don't think it's going to be as much work as it actually ends up being. It took about 8 of these gorilla cart loads worth just to fill one of the plots. And this would have never been able to occur if I hadn't had all of the help from the staff at Tyson Research Center, as well as all of the undergraduate students who worked with me. As filling those plots back up took about 3 months of work. And then I had to grow the seedlings of all 20 species in the greenhouse. And I inoculated them with the soil from Tucker and the old field at Shaw. And I planted them all in a grid in each of the plots. And so they are already inoculated at least 1 week prior to me planting them in the plot. And then finally, the growth metric that I took was harvesting the above ground biomass of each individual plant, and then drying and weighing each of those. So at this point, we just finished our 2nd harvest. I only have results for a little bit of the 1st year, but I'm hoping to have those results out soon. So the first objective for this project was to determine whether soil microbial origin, the remnant prairie old field soil, is important for the growth of these hard to establish tallgrass prairie plant species. So I expected for these hard to establish species to grow larger in the remnant soil, a little bit smaller in the old field soil, and then much, much smaller in the sterile soil. So what I've found so far, I only have results for 2 of these hard to establish species. Asclepias sullivantii, Sullivan's milkweed or prairie milkweed, it grew larger in the presence of soil microbes. You see old field and remnant soil, it grew bigger in those than in sterile. This effect was not significant. And this is growth after only 4 or 5 months after I established the experiment. So it's going to be interesting to see if there is a soil effect after I analyze the data from this year. Now for Antinaria neglecta, also known as prairie pussytoes, there was no evidence at all of a soil effect, at least in the first year. Now, my second goal was to determine the role of plant evolutionary history on the growth of hard to establish plant species. And my third one was to tease apart these contributions of the soil microbes from competition. So I expect that whenever the hard to establish plant species is growing in a community that is much more closely related to it, that it will grow smaller than if it is growing in a community where the background species are more distantly related to it. Now this could come about due to microbes or competition. So what's important about my experiment is that I have the sterile component to be able to tease apart the contributions of both of those. So again, if it is microbially determined, I expect for those plots that contain the soil microbes, the remnant prairie soil and the old field soil, that my plants that are much more distantly related to the background species would grow much larger. And that's specifically because they would be escaping those soil pathogens that would be attacking them. And then in the sterile soil, and that's the line that says without microbes, I wouldn't expect a phylogenetic effect at all. Now, if it's competitively determined, then I would expect for all of the plots, including the sterile ones, to have the same response. So the more distantly related plant species would grow much larger, while the hard to establish plant species would grow much smaller when they're in a community surrounded by those that are much closer related to them. So for Asclepias sullivantii, that is exactly what I found. I have not found a soil effect. Again, it's only 4 or 5 months after I started the experiment. But what's really exciting is that I found that this hard to establish species, whenever it is growing in a plot where the other species are much more distantly related to it, it grows much, much bigger than it does whenever the surrounding plants are more closely related. And this was true in all of the soil treatments. I found the same thing for prairie pussytoes, Antinaria neglecta. It wasn't significant. It was pretty close to it. So I'm really excited to be able to analyze my data from this year and see if this pattern holds true and whether there is a soil effect now after over a year of growth. So hard to establish plant species grow less whenever they're in communities that are more closely related to them. And I think that this is a really important result for people who do perform ecological restoration. If they have a plant species that they're having a really hard time getting to grow in the restoration, then perhaps they could come up with a seed mix that only possesses plants that are more distantly related to that plant species, and it would enable it to escape the competition with other species. So right now I'm going to be performing some DNA metabarcoding on this experiment as well. And I'm doing this with Dr. Christy Edwards and Dr. Bergen Bassner at the Missouri Botanical Garden again. And I want to determine the identities of the soil microbes, but I also want to look at differences in the soil microbial community composition. Okay, so finally, I'm going to get really specific and talk about the effects of soil microbes on the federally endangered Astragalus bibullatus. This species only has about 8 populations left on the planet, and they're all found in these limestone cedar glades within 10 square kilometers of Murfreesboro, Tennessee. If you look at this map here, you'll see that some of these populations, the circles with the X's through them, we knew they used to be there and they've gone extinct for various reasons. So part of my experiment, I wanted to look at the effects of soil microbes from communities where the species already is present, and then also places where we would like to perform a reintroduction in the future, and I'm calling those historically absent sites. So this is a legume species, it's in the bean family. So that means that it forms a symbiosis with bacteria called rhizobia. It forms these root nodules that provide nitrogen to the plant in exchange for carbon for the bacteria. And this is a really important symbiosis. It enables legumes to be able to thrive in soils that are really low in nitrogen. It also provides them with more defense against drought and herbivores. So this species, despite performing multiple reintroductions, there have been a few failures, unfortunately. And this is despite trying to match the reintroduction sites to the sites where the species already is growing. So they're trying to match it based on canopy cover, bryophyte cover, looking at the soil nutrients like nitrogen and phosphate. And they still were not able to get the species to grow very well in these glades. So I went ahead and went to Tennessee and collected soil and cover data from 9 different glades. 5 of them are glades where the species currently occurs, and 4 where we would like to reintroduce the species, because I wanted to gather evidence that these glades were not different from each other. And if you look at this principal components analysis that I performed, you see that the historically present glades where Astragalus bibullatus already is growing, they aren't really all that different from the historically absent glades that we would like to reintroduce them. So that leads me to think that soil microbes could be a really important factor for the reintroduction of this plant species. So my first objective was to determine whether those soil communities at these sites where the species already occurs, historically present glades and historically absent glades, would differentially affect growth of the species, Astragalus bibullatus, and its congener, Astragalus tennesseeensis, and the common grass Schizocarium scoparium. I also wanted to determine what the potential mechanisms are behind any growth differences. So I'm not going to go very in depth into the setup of this experiment because I already have explained a similar experiment, the first greenhouse one that I did. The big difference from this is that it was only 3 species this time, and instead of collecting soil from one site, I collected it from 9 different glades so that I could tell, so I could look at whether there is a difference between the historically present sites and the historically absent sites. And this was performed in live and sterile soil as well. So I expected for Astragalus bibullatus to grow significantly larger in historically present soil, but I only expected to see this in the live soil from the historically present sites. For the historically absent sites, I expected for it to grow in the live soil about the same as it grows in the sterile soil, because I expect for those sites to not possess the symbiotic microbes the species needs to be able to grow well. And that is exactly what I found. I found that it grows significantly larger in the presence of historically present microbes, but only in the live soil. So it was a very exciting result. And these data are currently in review at Restoration Ecology Journal. So my second objective for this study is to determine any potential mechanisms behind growth differences. So one thing that we did whenever we harvested the plants, we harvest the above ground biomass, but we also wash all of the roots from each of the plants. And while we're doing that, we count the nodules on each plant. So we were able to look at how many nodules Astragalus bibullatus had when it was growing in its own soil, when it was growing in Astragalus tennesciensis' soil, and in the grass's soil for both historically present and historically absent glades. So I expected to find that the species would have significantly more nodules whenever it was growing in Astragalus bibullatus soil in the historically present glades. And again, that is, well, I did find that it produced significantly more nodules whenever it was growing in soil from historically present glades. What's interesting is there also is a soil origin effect where it grows much larger whenever, or has more nodules whenever it is in the Astragalus bibullatus or Astragalus tennesciensis soil compared to that of the grass's soil whenever it's in historically present glades. And I don't find this result very surprising because I wouldn't expect for grass soil to have very many of the species-specific microbes that the species needs. But most importantly, I found that Astragalus bibullatus plants that possessed more nodules also grew significantly larger. So this gets at a potential mechanism behind why these plants grow so much larger whenever they are in the soil from historically present glades. So potentially, we need to explore this a little bit more in depth. It could be the rhizobial bacteria at the historically present sites are the ones that are species specific, but at the historically absent sites, those rhizobia are absent. And I need to definitely give a shout out to an undergraduate who worked on her senior thesis project with me. Kelly Showalter was integral to getting this experiment done. It was a huge experiment. She had an offshoot project for her senior thesis, but she spent hundreds of hours working on this project with me, along with finishing her senior year. And I am extremely grateful to all of the work that she's put in on this project. Thanks, Kelly. So Astragalus bibullatus did exhibit dependency on symbiotic microbes from historically present glades. And we do believe that they are likely to be rhizobia, but more experiments do need to be performed to pinpoint the identity of the microbes that are impacting the species. Currently, we are working to utilize DNA metabarcoding to determine the identities of soil microbes, whether specificity exists, and the differences in community composition based on site history. And I am, again, working with Christy Edwards and Bergen Bassner at the Botanical Garden to do this. We're looking at this in the soil, in the roots, and in the nodules of each of the plants. And finally, I think it's really important for those who are working to reintroduce this species to attempt to inoculate it with the species-specific rhizobia whenever they're doing future reintroductions. And this can be difficult because at this point, we don't have the ability to culture these specific rhizobia. So the most likely thing that can be done is to take the whole soil from those historically present glade sites. But care definitely needs to be taken when performing this kind of action, because if you take too much soil too frequently from a reference ecosystem, you have, you're much more likely to negatively impact it. Okay. Finally, I have a lot of people to acknowledge. Most importantly, I want to thank my partner, Seth Lindsay, for his constant support throughout my PhD, and many, many high school students from the Shaw Institute for Field Training. They helped out with the harvest of both of my greenhouse experiments. I definitely wouldn't have been able to do it without their help. Kelly Showalter, again, and I've had many undergraduate mentees through the Tyson Research Center over the summer, and these are just a few of them who have helped a lot on my experiments. Mike Dyer at the greenhouse at WashU, And the Missouri Master Naturalists have helped out with my research gardens at Tyson Research Center. I especially want to thank Bob Virag for his help. And Dr. Kim Medley and everyone at Tyson Research Center, Pete and Tim, for all of the help. And finally, I want to give my biggest thanks to my advisor, Dr. Scott Mangan, for his constant support throughout my PhD. And also our former postdoc, Dr. Claudia Stein. Okay, questions?
All right, thank you, Rachel, for that really phenomenal presentation of your research. Time for us to look at the first questions from the YouTube chat. And the first question was regards to one of your plant soil feedback experiments. And it was, did you try soil that had been exposed to each species and then sterilized to look for factors made by species A that benefit species B independent of microbes?
Yes, so there was a soil microbial, so we did have a sterile component of that. So I don't know if I should try to go back to the slides. I don't want to mess anything up. But I mean, first we trained or cultured the soil for each species, but it was very important to have that sterile component where we took the inoculum and sterilized it before putting it in the pots as well. So yes, that in that way, we're able to tease apart the impacts of perhaps any abiotic changes that came about because of having those particular plant species growing in the soil from the microbial impacts. Good question.
Hey, Rachel, great talk. Thank you so much. So we have another question. This is more of a statement, but I think it might be interesting food for thought, so I will ask it. Just sort of tying into that last question, Yusan wonders whether an alternative hypothesis based on doing the sterile switchover that was just asked, would it be that there is remaining nutrients left in the pot that was different due to niche differentiation?
Yeah, another great question. So whenever doing these plant soil feedback experiments, it's really important to make sure that you don't add too much of the soil inoculum. So we typically put less than 10% of the total soil that we put in the pot is the soil inoculum. In my case, I put only about 8% of the soil was the soil inoculum. And there have been studies out there that have demonstrated that there is no abiotic effect whenever you do put such a small percentage of the soil inoculum in there. Good question.
While we wait for additional questions to populate the chat, I have a question, and that is that on your question slide here, you have an array of pollinators on display. And have you collected any data on the pollinators in your mesocosms at Tyson?
Unfortunately, not yet. I would love to. And my plan was to do that with undergraduates this summer, and COVID kind of put the brakes on that. But if I do end up taking students this summer at Tyson, that would be something I would love to have people look at. I do want to point out the adorable jumping spider in the Asclepias incarnata flowers in the bottom right-hand corner. Yeah.
Excellent. So multiple guilds of invertebrates.
Right. We have had one student, Kit Lord, who looked at the different, I guess, guilds of spiders in the experiments.
Yeah, I was wondering, were there additional students who carried out projects on other species besides the one you just mentioned? It seemed like such an amazing design for you.
You mean beside the Astragalus?
Yeah.
We had one student, Reagan Rainey. She decided that she wanted to look and see if mycorrhizal fungi were really important for the hard to establish species. For anyone who doesn't know the importance of mycorrhizal fungi, about 80 to 90% of land plants form a symbiosis with mycorrhizal fungi. And these fungi infect the roots of plants, and they provide a lot of benefits, like giving them phosphorus, protecting them from viruses, increasing absorbance of water from the soil in exchange for carbon. So it's a very important symbiosis. So Reagan spent a lot of time over the summer growing these species and the different inocula that I used, staining the roots with trypan blue and then mounting them on slides. Unfortunately, with that kind of experiment, she didn't have enough time to grow the plants as long as you probably needed to actually see an effect of the mycorrhizal fungi, but it was very cool that she did that.
I'll ask another question. So if you had a huge amount of money and a huge team of students and a whole fleet of excavators and people operating those excavators, and could have expanded your mesocosm experiment even further, what is an example of one type of treatment that you would think would be really interesting to add on to that experiment?
Oh man.
I'm not saying you should do this as one of your dissertation committee members, please don't. But
I'm done, I promise. I don't know about treatment. I think that the big thing that I would like to do now, especially in a postdoc, hint, hint, I'm looking for a postdoc. I would like to move this to the field. While going from a greenhouse to a research garden does make it a little bit more close to what you would expect in nature, it is still in a much more controlled environment than you would experience if I would have dug these holes out in a field and performed this experiment. So I would like to be able to perform, I guess you'd say, more realistic experiments at even a larger scale.
It looks like we have some more questions coming in the chat now.
Yeah.
So one question from one of our newer PhD students in the program, Eli Callison, is, can you tell us about how you came to study this? What was the process like in developing the various parts of this project?
I'm guessing we mean like my whole PhD.
Right, but sorry, the question was prefaced by, this is a really impressive PhD thesis, which it is.
Thank you. So when I was an undergraduate, I worked with Dr. Matthew Albrecht from the Missouri Botanical Garden, and I had a senior thesis project looking at the effects of shade on Astragalus bibullatus's growth. And it got me wondering whether soil microbes could be really important for this species. And that's how I actually ended up meeting my advisor, Dr. Scott Mangan, because I sent him an email and I met with him in his office, and then like the PhD kind of snowballed from there. But I really put out a lot of effort to try to tailor my dissertation to be something that I cared about, that I was passionate about. I could have probably just pretty easily gone along with what my advisor would have preferred, but thankfully he did give me the freedom to explore the questions that I'm really interested in and care about. And having partners at the Missouri Botanical Garden was essential. I couldn't have done the Astragalus bibullatus experiment or any of the DNA metabar coding without them.
Collaborations are very important.
Yeah.
We have several questions coming in here. Barrett Terrier would like to know, I've read that some plants can recognize related plants and grow slower so that they don't outcompete them. Do you think that this communication might make factor into your experiment? And then they add, I'm not 100% sure of the credibility of this claim.
I could see that being a factor, perhaps through mycorrhizal fungal networks. It has been shown that plants are connected to each other under the ground by mycorrhizal fungi. Because my experiment, especially like the Astragalus bibullatus, was performed only with individual plants, I'm not able to actually look at that right now, but it could be possible.
Okay, we have another question here. Or a person says, "Yay, yay, Kelly." Would you recommend that a restoration remove related species in order to establish the desired HTE species?
Oh man, that is a really good question. I know that a lot of restorations are performed in places like an old field, which is why I selected soil from an old field at Shaw Nature Reserve. But those old fields do have a mixture of native plants as well as invasive, non-native species. I think that that could be a good recommendation, but you do have to be careful. Like maybe if those species are ones that are also hard to establish, I wouldn't want to mess with those as well. So a lot of care needs to be taken whenever you're doing a restoration and taking these factors into account. Yeah, we've had a discussion. We have a Soil-Microbe and Restoration Journal Club, and we've talked about the different kinds of restorations. And if you're trying to improve upon, say, like a remnant prairie, you wouldn't want to be manipulating that very much because you don't want to destroy or impact the species too much. But if it's an old field that used to be like a cornfield or a wheat field or something like that, I think it's much more reasonable to be manipulating the plants that are there right now.
Another question about the applied aspects of your work is whether you see possible field applications for this in smaller landscapes, such as green roofs or urban infrastructure reuse projects, both of which usually use engineered soils.
Yeah, I think it could be really important. I mean, especially with agriculture, if you're trying to grow a garden with vegetables, we know that like we do crop rotation because of soil microbes. The traditional explanation is that like nutrients are used up in the soil and then you need to grow a legume to replenish the nitrogen in the soil. But what's mainly happening is that if you're growing, say, a big monoculture of wheat, over time, you're having the pathogens building up and it starts suppressing the growth of that monoculture of plants. And so you need to give that field a break and allow other microbes to accumulate in the field. And I could see that being the same for a green roof or something like that, growing plant species that are maybe a little more distantly related to each other in the same plot would potentially increase their growth or increase their yield if you were trying to grow vegetables or something like that.
All right, we have one final question here for you. Do you think your results, especially on evolutionary relatedness, may inform why invasive or alien congeners of native species are particularly problematic in native ecosystems?
Yeah, I think that that could be a factor. We are finding that it's the enemy release or pathogen release that's happening. So just think like, I don't know, bush honeysuckle or some other species coming here from another country, it's escaping its pathogens. And we do have other honeysuckle species here, but it just doesn't have the species-specific pathogens that attack that particular species. More research really does need to be done on this, but I do know that there are several labs that are particularly studying the impacts of relatedness and enemy release.
Okay, Rachel, thank you so much for that really fantastic overview of your super impressive dissertation research. There would be thunderous applause if we were all together in person, but hopefully you can hear some of that thunderous applause with our LEC Biodiversity Fellows at the Zoom reception that will begin in just a few minutes.
Yeah, thank you to everyone for all the great questions. I really enjoyed speaking with you.